This is Module 313 of the online course on genetic selection from concepts to application to genetic improvement of health. My name is Jack Deckhurst from Iowa State University, and I will be the presenter for this module, which focuses on genetic improvement of quantitative disease traits, in other words, disease traits that are affected by many genes as well as by environmental factors. In this module, I will discuss approaches for the collection of phenotypes that can be used to select for disease resistance. I'll discuss the concepts of resistance, tolerance, and resilience. I'll discuss whether selections should be for specific diseases or for general robustness or resilience. And I'll present a natural challenge model that has been established to identify the genetic basis of resilience in pigs. To target your learning, I'd like you to keep the following questions in mind as we go through this material. First, how can breeders select for disease-related traits in nucleus herds that must be kept disease-free? Do animals that disease-related phenotypes are collected on need to be close relatives of selection candidates for those phenotypes to be useful in selection of those selection candidates? Third, should we select for resistance, tolerance, or resilience? And finally, should the focus be on specific diseases, or should it be on general robustness or resilience to multiple diseases? One of the big challenges for the incorporation of selection for disease resistance in breeding programs is that it is very difficult to obtain relevant phenotypic data that can be used to estimate breeding values for disease resistance on selection candidates in nucleus herds. The reasons for this include that there are often no clear and easy to record phenotypes, that recording or diagnosis of diseases is often inconsistent, especially across farms, and in addition, selection candidates are often kept in facilities with high biosecurity, which means that the disease data cannot be recorded on selection candidates. Examples are AI bulls, which must be kept disease-free to allow marketing and export of their semen, and the high-health nucleus farms that are used in pig breeding programs, again in order to enable sale of germ stock from these farms, both domestically and abroad. Another challenge for genetic improvement of disease traits is that most disease-related phenotypes have low heritability. And finally, care must be taken that selection for resistance to one specific disease may make animals more susceptible to other diseases. There are several approaches that can be used to overcome at least some of these challenges. One is to develop disease-indicated traits that can be collected on selection candidates without having to expose them to disease, and I will give a couple of examples of such indicated traits. Another approach is to collect phenotypes related to disease in commercial farms and then utilize that information to estimate the breeding value of selection candidates in nucleus herds. And finally, health-related phenotypes can be collected in special experimental facilities, followed by the use of that information for selection in the nucleus. Phenotypic data on disease-related traits that is generated with these approaches can be used for selection using one of three approaches, which we have already described in previous modules. First, they can be used to estimate breeding values related to disease on selection candidates using the collected phenotypes. Second, by identifying major genes or genetic markers that are associated with disease, and then using those to genotype and select selection candidates. And third, by using genomic prediction using high-density SNP genotypes. A prime example of using an indicated trait for selection for disease resistance is somatic cell count in milk as an indicated trait for mastitis in dairy cattle. Whereas records on incidence of mastitis are often incomplete and inconsistent, and they only capture clinical cases of mastitis and also have low heritability, Somatic cell count captures both clinical and subclinical mastitis, is more heritable, and is easily recorded on milk samples that are routinely submitted for milk recording. Using this data, genetic evaluations for somatic cell count were implemented in the US in 2000, which allowed dairy producers to select bulls with lower somatic cell count, which has resulted in the decline in somatic cell count that you see in this figure, and an associated reduction in the genetic susceptibility of cows to mastitis. Another example in dairy cattle is the high immune response technology that was developed by Dr. Bonnie Mallard from the University of Guelph in Canada. This was developed for dairy cattle, but is now also being invested by that group for use in pigs. High immune response technology involves two tests that can be performed on healthy animals, including bulls, that capture an animal's ability to resist a wide array of diseases by measuring the animal's potential to generate a cell-mediated response and also antibody-mediated response. To measure cell-mediated response, a skin test is performed by injecting an antigen that drives this type of response into the tail fold. This picture on the left shows measuring the tail folds to determine the cell-mediated response. The side measured here is the control side, whereas the other side is the test side. The picture below shows the infiltration of cells at the injection site to cause swelling at that site, with all the purple dots being immune cells. To measure antibody-mediated response, blood samples are taken after exposure to a pool of antigens, and the antibody activity is measured using an ELISA test, with more antibody activity indicating a higher antibody response. Results in this bar graph show that on average, cows that score high for both of these tests have less disease, showing the value of these immune tests. High immune response technology for dairy cattle is being used by CMEX Canada under an exclusive license from the University of Guelph, and bulls that score high on these tests are marketed as being Immunity Plus. As I mentioned, similar tests are currently being evaluated for use in pigs, which would allow immune response to be measured on pigs in a biosecure nucleus environment as an indicator for susceptibility to disease in the field. Another strategy to obtain information on disease-related traits or on performance under disease is to record disease-related phenotypes on commercial animals in the field. So for reproduction-related traits, phenotypes would be recorded on sow herds, and for grow-finish-related traits, disease phenotypes would need to be recorded on finisher pigs. These phenotypes would then be used for genetic evaluation of animals in the nucleus herds. A major challenge with this approach, however, is that apart from the logistics involved in collecting this data, it would require pedigrees to be tracked through the system, because in order to compute an EBV of a nucleus animal for a trait that is recorded in the field, you need to have phenotypes on relatives. Recording pedigrees on commercial animals, however, is a big challenge, especially with the use of mixed semen. In addition, there will be a time delay in getting information on relatives from the field, especially if you wait for phenotypes on progeny. This would increase generation intervals and then reduce response per year. Nevertheless, this is an approach that has been implemented in a number of breeding programs in pigs, allowing selection more directly for performance in the field rather than performance in a high health environment. Another strategy for utilizing phenotypes for selecting breeding stock in the nucleus is genomic prediction. As discussed in a previous module, the general idea behind genomic prediction and genomic selection is to genotype a substantial number of animals that have recorded phenotypes for traits of importance, and to genotype them using a high-density SNP chip. Using these phenotypes and genotypes, the prediction model is then trained by estimating the effect of each SNP and using the resulting estimates to compute the genomic EBV of selection candidates after they have been genotyped using the SNP chip. In this case, the training population would consist of genotypes and phenotypes collected on crossbred animals in the field, and the selection candidates would be the animals in the nucleus. So data and high-density SNP genotypes would be collected on crossbred pigs in the field, on crossbred market pigs for the grow-finish phase, and on crossbred sows for the reproduction phase. To focus on disease-related traits or performance under disease, the farms that these data are collected on should have some history of disease pressure. The resulting genotypes and phenotypes would then be used to estimate SNP effects for disease and performance in the field through genomic training, which would then be used to predict the breeding value of nucleus animals for field phenotypes based on the high-density SNP genotypes of selection candidates. This would allow genomic selection of nucleus animals for disease and performance in the field. One important difference of this genomic prediction approach compared to the strategy where phenotypes collected in the field are directly used for genetic evaluation in the nucleus is that the genomic prediction approach does not require pedigree to be tracked through the system. In addition, the animals that are phenotyped do not need to be close relatives of selection candidates, although accuracy of genomic prediction would be higher if they are close relatives. Another strategy is to obtain disease-related phenotypes in experimental facilities. This approach has been used by Highline International to improve survival to Marek's disease in layer chickens. In their system, each generation, progeny of male selection candidates are infected with the latest, most virulent field isolate of Marek's disease, and the percent mortality is recorded, and then used to evaluate roosters for survival to Marek's disease. In this system, phenotypes recorded on relatives are fed back to the nucleus and then used to estimate the breeding values of selection candidates for these disease-related traits. Another approach that can be used here is to genotype individuals that are challenged using a high-density SNP chip and then use the resulting SNP effect estimates for genomic prediction and genomic selection in the nucleus. Similar to the approach of genomic selection using commercial crossbred data that is collected in the field. Again, the advantage of the genomics approach is that the individuals that are challenged do not need to be close relatives of the selection candidates, and pedigree is not needed. This is the approach that was used in the PRRS Host Genetics Consortium, which we will discuss in Module 4, in which large numbers of weaner pigs from commercial lines were challenged with the PRRS virus to identify genomic regions and genetic markers that are associated with resistance to PRRS virus infection, which could then be used for marker-assisted selection or genomic selection in nucleus breeding programs. One of the questions when selecting on disease-related traits is whether selection should be for resistance, tolerance, or resilience or robustness. As discussed in a previous module, resistance refers to the ability of the animal to prevent infection or limit replication of the pathogen once infected. So at a given level of exposure, individual A in this example has a lower pathogen load, for example, as measured based on the level of viremia in blood, than animal B, and therefore animal A is more resistant. Tolerance is defined as the ability of the animal to maintain productive performance at a given pathogen load. This is represented by the slope of the regression of performance on pathogen load. In this example, animal B is more tolerant than animal A because its growth rate does not decline as fast as pathogen burden increases compared to animal A. Resilience or robustness is the combined effect of resistance and tolerance and refers to the ability of the animal to maintain performance upon exposure to a pathogen. Thus, although animal B is more tolerant as pathogen burden increases, animal A has a better resilience because its performance drops less as level of exposure increases. This is the net effect of the greater resistance of animal A despite its lower tolerance. Whether animal A performs better in a given environment than animal B then depends on the difference between A and B in performance in a clean herd, which is the intercept of these lines in the resilience graph. It also depends on their difference in resilience, in other words, the slope of performance on level of exposure, and on the level of exposure. Based on this, using 0.75 as the level of exposure, animal A would be preferred because its performance at that level of exposure is better than that of animal B. And you can see that in the tolerance graph, where for those levels of exposure and the associated pathogen loads, animal A has a higher growth rate than animal B. And you can also see that directly in the resilience graph, where for a level of exposure of 0.75, animal A has higher growth rate. However, for lower levels of exposure, animal B would be the preferred animal. Note that the difference of performance at exposure level 0 is the difference of performance that is evaluated in a biosecure nucleus herd, but this may not result in selection of the right animals for performance in the field, as illustrated by these graphs. Estimating resistance, tolerance, and resilience of animals is one of the current challenges in animal breeding. These concepts of resistance, tolerance, and resilience are defined for each infectious pathogen. Of course, in the field, animals are exposed to multiple pathogens, often at the same time. So to investigate the genetic basis or resilience to multiple diseases, a large collaborative project was initiated 2 years ago on the application of genomics to improve disease resilience and sustainability in pork production. This project is led by Michael Dick from the University of Alberta, along with John Harding from the University of Saskatchewan and Bob Kemp from PigGen Canada, and myself, who is involved primarily in data analysis. This project is financed by Genome Canada, Genome Alberta, and Genome Prairie, as well as a grant from USDA NIFA, and involves collaborations from multiple universities and from members of the PigGen Consortium, which is a consortium of breeding companies in Canada. An important component of this project is the establishment and use of a natural challenge model to characterize and quantify the genetic basis of resilience to disease in grow-finish pigs. To that end, a research station of CDPQ in the province of Quebec in Canada has been used as a natural challenge facility, which has been seeded with these pathogens by bringing in seeder pigs from farms with known disease. These seeder pigs then have been mixed with Yorkshire Landrace weaner pigs from high health herds in batches of 60 to 75 pigs every 3 weeks in a continuous flow system, so a mixing of old batches with incoming batches. Before these pigs go into the natural challenge facility, they enter a quarantine nursery where they are kept for 3 weeks. These 3,500 Yorkshire Landrace weaner pigs come from 7 industry partners from PigGen Canada that are collaborating in this project. In the fall of last year, the following pathogens were present in the natural challenge facility, including PRRS, influenza, PCV2, along with a series of bacteria that are common in industry. Jointly, this represents a substantial disease pressure, which probably is higher than typically seen in the field. However, it allows for clear expression of genetic differences in resilience to disease. The project has careful veterinary oversight to ensure proper care of animals. In the Natural Challenge facility, the following phenotypes are collected, which are being used to quantify resilience: body weight every 3 weeks, daily feed intake, water intake, mortality and morbidity, of course disease diagnostics, health treatments, and as well on a small number of animals, the gene expression level in blood is being evaluated, as well as the gut microbiome based on fecal samples. In the quarantine nursery, we are collecting a number of samples and conducting a number of assays with the aim to identify biomarkers that could be collected on young healthy pigs and that are potential predictors of resilience to disease. This includes several immune response measures, including the high immune response test developed by Bonnie Ballard of the University of Guelph, as well as several in vitro immune tests and phagocytosis tests. We will also, on 1,000 pigs, quantify the whole blood transcriptome at a young age while they're still healthy, as well as a large number of small metabolites in the blood. and quantify the blood proteome, and also analyze the gut microbiome based on fecal samples. All pigs that go through this system will be genotyped using a high-density SNP chip, which will allow us to get 650,000 SNP genotypes across the genome. And these data will then be integrated through statistical and bioinformatic analyses with the aim to identify phenotypic predictors of resilience and genetic predictors of resilience based on data that is collected while they are young and healthy, similar to what would be obtainable in nucleus herds. And we will also be able to identify trait genetic parameters, architecture, and mechanisms related to resilience of disease. So in summary, genetic improvement of resistance or susceptibility to quantitative complex diseases can be achieved by selecting on disease indicator traits that are collected on selection candidates or their relatives, or by selecting on EBV or genomic predictors of disease-related phenotypes that are collected in the field or that are collected in experimental challenge facilities. In all these cases, genomics increases the potential impact of these strategies for genetic improvement in animal health because it allows us to leverage these phenotypes across a larger number of animals. And finally, genetic improvement of disease probably needs to focus on robustness and resilience against multiple diseases rather than on resistance to individual diseases. So this concludes this module, and I will now direct your attention to the questions that were posed earlier, and I hope you can now answer those based on the material provided. Thank you for your attention.